Hello, welcome to Biology, the scientific study of life. Individual bacteria respond to environmental change by regulating their gene expression. Mutations and various types of gene transfer generate the genetic variation that makes natural selection possible. And natural selection acting over many generations can increase the proportion of individuals in a bacterial population that are adapted to some new environmental condition. But how can an individual bacterium looked into the genome it has inherited copy with environmental fluctuation? Consider, for instance, an individual E. coli cell living in the erratic environment of a human colon, dependent for its nutrients on the whimsical eating habits of its host. If the environment is lacking in the amino acid tryptophan, which the bacterium needs to survive, the cell responds by activating a metabolic pathway that makes tryptophan from another compound. Later, if the human host eats a tryptophan-rich meal, the bacterial cell stops producing tryptophan, thus saving itself from squandering its resources to produce a substance that is available from the surrounding solution in a prefabricated form. This is just one example of how bacteria tune their metabolism to changing environments. Metabolic control occurs on two levels. Notice this figure, this one level regulation of enzyme activity, and this one regulation of enzyme production. First, cells can adjust the activity of enzymes already present. This is a fairly fast response, which relies on the sensitivity of many enzymes to chemical cues that increase or decrease their catalytic activity. For example, the activity of the first enzyme in the tryptophan synthesis pathway is inhibited by the pathway's end product. Thus, if a tryptophan accumulates in a cell, it shuts down the synthesis of more tryptophan by inhibiting enzyme activity. Such feedback inhibition, typical of anabolic biosynthetic pathways, allows a cell to adapt to short-term fluctuations in the supply of a substance it needs. Second, cells can adjust the amount being made of certain enzymes. That is, they can regulate the expression of the genes encoding the enzymes. If, in our example, the environment continues to provide all the tryptophan the cell needs, the cell stops making the enzyme that work in the tryptophan pathway. This control of enzyme production occurs at the level of transcription, the synthesis of messenger RNA coding for these enzymes. More generally, many genes of the bacterial genome are switched on or off by changes in the metabolic status of the cell. The basic mechanism for this control of gene expression in bacteria, described as the Oberon model, was discovered in 1961 by Francois Jacob and Jacques Monod at the Pasteur Institute in Paris. Let's see what an operon is and how it works, using the control of tryptophan synthesis as our first example. Okay, notice also the regulation of metabolic pathway. In the pathway for tryptophan synthesis, an abundance of tryptophan can both inhibit the activity of the first enzyme in the pathway, feedback inhibition, a rapid response, and B also represent expression of the genes for all the enzymes needed for the pathway, a longer term response, and the minus here, notice the red cycle here, simple stands for inhibition. Oberon's the basic concept. E. coli synthesizes tryptophan from a precursor molecule in a series of steps. Each reaction catalyzed by a specific enzyme. The 5 genes coding for this enzyme are clustered together on the bacterial chromosome. A single promoter serves all 5 genes, which constitute a transcription unit. Recall from chapter 17 that a promoter is a site where RNA polymers can bind to DNA and begin transcription. Those transcription gives rise to one long mRNA molecule that codes for all 5 enzymes in the tryptophan pathway. The cell can translate this mRNA into 5 separate polypeptides because the mRNA is punctuated with start and stop codon that signal where the codon sequence for each polypeptide begins and ends. A key advantage of grouping genes of related function into one transcription unit is that a single on-off switch can control the whole cluster of functionally related genes. When an E. coli cell must make tryptophan for itself because the nutrient medium lacks this amino acid, all the enzymes for the metabolic pathway are synthesized at one time. This switch is a segment of DNA called an operator. Both of its location and name suit its function, positioned within the promoter or between the promoter and the enzyme coding genes. The operator controls the access of RNA polymers to the genes. Altogether, the operator, the promoter, and the genes, they control the entire stretch of DNA required for enzyme production for the tryptophan pathway constitute an operon. Here we are dissecting the trip operon, trip for tryptophan, shorter. One of many operons in the E. coli genome. In this figure, what's that? If the operator is the switch for controlling transcription, how does this switch work? By itself, the trip operon is turned on. That is, RNA polymers can bind to the promoter and transcribe the genes of the operon. The operon can be switched off by a protein called the repressor. The repressor binds to the operator and blocks attachment of RNA polymers to the promoter, preventing transcription of the genes. A repressor protein is specific, that is, it recognizes and binds only to the operator of a particular operon. The repressor that switches off the trip operon by binding to the trip operator has no effect on other operons in the E. coli genome. The repressor is the product of a regulatory gene called TRPR, which is located some distance away from the operon it controls and has its own promoter. Regulatory genes are expressed continuously, although at a low rate. And a few repressor molecules are always present in E. coli cells. Why then is the trip operon not switched off preeminently? First, the binding of repressors to operators is reversible. An operator vacillates between two states. One without the repressor bound and one with the repressor bound. The relative duration of each state depends on the number of active repressor molecules around. Second, the trip repressor-like mass regulator proteins is an allosteric protein with two alternative shapes, active and inactive. Notice this one. Bo. The trip repressor is synthesized in an inactive form with little affinity for the trip operator. Only if tryptophan binds to the trip repressor at an allosteric site does the repressor protein change to the active form that can attach to the operator, turning the operon off. Tryptophan functions in this system as a copressor, a small molecule that cooperates with a repressor protein to switch an operon off. As a tryptophan accumulates, more tryptophan molecules associate with the repressor molecules, which can then bind to the operator and shut down the production of the tryptophan pathway enzymes. If the cell's tryptophan level drops, transcription of the operon's genes resumes. This is one example of how gene expression responds rapidly to change in the cell's internal and external environment. Notice also this figure that operon regulates synthesis of reversible enzymes. Tryptophan is an amino acid produced by an anabolic pathway catalyzed by reversible enzymes. Notice that here tryptophan 5 pathways are grouped along with a promoter and an operator into pathway are grouped along with promoter and an operator into the trip operon. The trip operator is located within the trip promoter. Be a combination of tryptophan, the end product of the pathway represses transcription of the trip operon, thus blocking synthesis of all the enzymes in the pathway. The mechanism in E. coli is shown here. Back to the tryptophan absent, repressor inactive, operon on. Notice RNA polymers attaches to the DNA at the promoter and transcribes the operon's genes. Notice that. Notice here promoter, trip RNA, regulatory gene, mRNA, closer look protein, inactive repressor. Notice also promoter here, RNA polymers, operator, closer look trip here, notice that. Start codon, stop codon, notice that. These are polypeptides that make up enzymes for tryptophan synthesis. These are, notice that. Tryptophan present, repressor active, operon off. As tryptophan accumulates, it inhibits its own production by activating the repressor protein. Notice that. No RNA, DNA, closer look DNA, mRNA, protein, and notice active repressor, no RNA made here. Okay, thanks for listening.